Nearly all membranes in living organisms have an electrochemical potential, meaning the concentration of molecules, ions, and charges differs across the membrane. This is naturally the case because the phospholipid bilayer of the membrane is semi-permeable, and only hydrophobic molecules such as oxygen or carbon dioxide, or very small polar molecules such as water, can freely cross the hydrophobic layer of the membrane. Ions and larger polar molecules need to be transported or pumped across the membrane. The kind and the number of pumps and transporters inside the membrane defines how the concentration of compounds differs across the membrane. This concentration gradient results in an excess of positively charged ions on one side of the membrane, and the electrochemical gradient can be used to power different processes. The plasma membrane potential in animal cells serves two main purposes. Firstly, it powers many processes such as transport of certain molecules into and out of the cell by coupling this process to the energetically favorable flow of another molecule or ion down that one's concentration gradient. For example, we can harness the high concentration of sodium ions outside the cell to actively transport glucose into the cell using a symporter that couples their transport. The passive transport of sodium down its concentration gradient powers the active transport of glucose against its concentration gradient. The second main function of the membrane potential is signal transmission within a cell or from one cell to the next. This is used in electrically excitable cells, such as neurons and muscle cells. In order to explain this electrical transmission of signals, we will first look at the distribution of ions across the plasma membrane of neurons and muscle cells when there's no ongoing signal transmission, meaning the membrane is at a relatively stable potential called the resting potential. Most animal cell plasma membranes have a negative resting potential, meaning the inside of the cell is more negative as compared to the outside of the cell. This resting potential is around negative 70 to negative 80 millivolts in neurons, and it is generated mostly by the sodium-potassium pump. It's a P-type pump and ATPase that transports sodium out of the cell and potassium into the cell, and it consumes ATP while doing so. Therefore, there is a chemical gradient across the plasma membrane. But in addition to the unequal concentration of ions, there's also an unequal concentration of electric charges, because the sodium-potassium pump transports 3 sodium cations out of the cell, while it transports only 2 potassium cations into the cell. So it's said to be electrogenic. This lack of positive charges inside the cell causes an electric gradient where the inside of the plasma membrane is negatively charged relative to the outside of the cell. There are also some potassium leak channels that are always open. So potassium flows out of the cell, driven by its concentration gradient that is higher inside the cell. But this facilitated diffusion does not completely equalize the work of the sodium-potassium pump because of electrostatic repulsion. The excess of the positive charges on the outside of the cell impedes the facilitated diffusion of enough potassium to equalize the chemical potential. So it is this balance, the constant action of the sodium-potassium pump and also the passive diffusion of potassium ions, which results in the negative resting potential. But there are also far more ions that play a role, so I will just leave them out here for simplification. But just so you know, for example, chloride ions are more concentrated outside the cell, but overall the concentration of anions, of negatively charged ions is much higher inside the cytoplasm. So the resting potential is quite complex, but it's most important that you understand the role of sodium and potassium. Now to signal transmission. An action potential occurs if an electrical stimulus depolarizes the membrane, meaning it makes the membrane potential less negative, so that the potential is higher than or around negative 50 or negative 55 millivolt. This threshold potential needs to be overcome because it is sensed by voltage-gated sodium channels. These channels were closed at resting potential, but when the membrane threshold potential is reached, they open. A positively charged alpha helical domain within the channel is repelled by the new positive charges in the cytoplasm, and it relocates within the channel towards the extracellular space. This conformational change opens the voltage-gated sodium channel, and this alpha helix remains in the open conformation until the membrane potential is lower than negative 55 millivolt again. So the open sodium channels allow the inflow of many sodium ions from the high concentration outside the cell through this channel into the cytoplasm. This depolarizes the membrane, raising the membrane potential to around positive 40 millivolt. As you were probably thinking already, this inflow of positive charges triggers the neighboring voltage-gated sodium channels to open, resulting in signal propagation. This signal cannot travel backwards, because at the site where the stimulus was first received, the sodium channels are quickly inactivated. There is a special channel inactivating segment attached to the channel. Soon after the channel opens, these segments plug the channels. This inactivation is not voltage dependent, but simply based on diffusion and affinity, so it occurs shortly after the channel opens. This plugging marks the peak of depolarization, because from now on, no more positive charges can enter the cell. The absolute refractory period begins, because during this time, no voltage-gated sodium channels can open, meaning no stimulus can be sensed. Around the same time, another kind of channel opens, a voltage-gated potassium channel. These channels were triggered by the same stimulus that opened the sodium channels, but they just took longer to open. So the opening of these potassium channels allows positively charged potassium ions to diffuse out of the cell, resulting in repolarization, because the cytoplasm slowly becomes more negatively charged again. As soon as the threshold potential is reached, remember, the alpha helical domain inside the voltage-gated sodium channel relocates towards the more negative cytoplasmic side. This closes the channel completely, and it pushes the channel inactivating segment out of the channel pore. This marks the beginning of the relative refractory period. Now that the sodium channels are not anymore inactivated, they can theoretically open again when a stimulus arises. However, the opening of the potassium channels has resulted in an excessive outflow of potassium ions, and this is called hyperpolarization. Now the membrane potential is even more negative than it was at resting potential because of this overshoot. Therefore, a much stronger stimulus is needed during the relative refractory period to initiate a new action potential. The sodium-potassium pump, meanwhile, and the potassium lead channels restore the resting potential, which concludes the action potential. This action potential travels down the axon of the cell, and the signal transmission is speeded up by myelination. This is an insulating sheath that wraps around most of the neuronal axon, and it allows only channels to be accessible at so-called nodes of round VA. The positive charges therefore do not need to diffuse inside the cell from one channel to the next. Instead, so-called saltatory transmission occurs because a charge is transmitted faster than ions diffuse inside the cell. When the signal reaches the axon terminal, it can then be transmitted to a neighboring neuron or muscle cell through cell junctions called synapses. If cells are directly linked by channels and the ions directly diffuse through gap junctions into a neighboring cell, this is called an electrical synapse. It immediately results in an action potential in the neighboring cell. It is very fast, but it cannot be regulated. So, there's also a chemical synapse, where the depolarization opens voltage-gated calcium channels. Calcium then rushes into the cell and binds to synaptotagmin proteins. These are localized at the plasma membrane. Calcium-bound synaptotagmin then allows the binding of snare proteins, which will make the synaptic vesicles fuse to the presynaptic membrane. Neurotransmitters from within the vesicle are released into the synaptic cleft, where they diffuse to the neighboring cell and activate it. The neurotransmitters, for example, can bind to ligand-gated sodium channels. This triggers their opening, and the depolarization occurs in the neighboring cell. So a new action potential is generated in the postsynaptic cell. As you can see, the chemical synapses are taking much longer than the electrical synapses, but they have the advantage that they can be regulated. So as you can imagine, this video greatly simplifies the entire process. So if you easily understand the content, maybe pick up a textbook and learn about the additional ions and transporters involved in the membrane potential and signal transmission, or about excitatory and inhibitory signals that are summed at the axon hillock, or even look into the chemical principles behind the membrane potential. Thanks so much for watching, and please comment any feedback and questions below.